Hey guys, welcome back to a new video by Biology Resumption. So today we are back with another past year paper video for the Cambridge IGCSE May-June 2024 series, the 0610 paper 4 variant 2. So like any of my usual past year paper videos, feel free to put your questions down in the comment section below, and also I will attach the past year paper link in the comment section below. So let's start off with this paper. So question 1. So fungal cells and plant cells, they contain mitochondria. Okay, why is that? Because they are eukaryotes. First thing you need to know that these two, they are the eukaryotes. It means that they have the nucleus. And then you need to state the function of the mitochondria. So the answer for this is aerobic respiration. The reason you need to specify is the term aerobic. This is very important because respiration on its own can either mean it's aerobic respiration or anaerobic respiration. And then you'll be asking me, why is it aerobic respiration mainly? Because a lot of the processes that occur in the mitochondria is mainly cater for aerobic respiration because oxygen is important. Okay. Part 2. State one feature, okay, that is used to distinguish a plant from a fungi. Okay. So your plant, you can always remember, is like one is a flower, okay, with leaves. Another one is a fungi, which is like a mushroom. So you can see that a feature can either be the difference of their cell wall, okay, and then another one is the presence of the chloroplast. So for this case, I've mentioned that the plant cell wall is made of cellulose, and then for the fungi is made out of chitin. Okay, chitin. And another feature that I mentioned was the presence of the chloroplast. So there's only chloroplast in the plant, but no chloroplast in the fungi. So that's pretty much it. And part B, yeast is a fungus that can respire to produce ethanol. So state the balanced chemical equation for the type of respiration in yeast. So for this type of question where they ask you for a balanced chemical equation, you have no choice but to memorize this if you did not take chemistry. If you did take chemistry, you just basically remember is basically from glucose they produce ethanol, and then you will get CO2. Ethanol, you know, is a CH3 CH2 OH. So you can just make it as C2H5OH and then plus CO2. Balance up the equation, make sure the left hand side is equal to the right hand side, then you are set. This is the 2 marks you're going to get. Part C, a scientist investigated the effect of sugar on respiration on yeast cells. So this would be respiration. So one flask contained 100 centimeter cube of sugar solution and another flask that contained 100 centimeter cube of water. Both flasks contain the same mass of yeast. So the temperature was maintained at 25 degrees. So this is at room temperature. So you can see that there's a thermometer to measure to ensure that there is 25 degrees constant, and you can see a yeast in the sugar solution. So you know that there's yeast and then there is glucose. And the result of it is to collect the carbon dioxide. Okay. So this is roughly how is it going to look. Then if you look at the next one, they say figure 1.2 is a graph showing the results of the investigation. Then using the gradient shown in figure 1.2, you just have to calculate the rate. So the rate is always the volume produced equals over time. Okay, volume of gas, and it will be over time. So you can just, they have mentioned that they want you to use the yeast in the sugar solution. Okay, this is the main one. So you'll be seeing this line. Okay, so you're going to get 10 and 15. 10 and 15 is roughly 11 here and 19. And you use final minus initial divide by the time. So it has to be precise. The only answer can be accepted is 1.6 centimeter cube. So you have to read exactly. So there is no further decimals. There's no other answers than just 1.6 centimeter cube per minute. So if you're confusing how to get the unit, so you know that is between volume is cm cube divided by minutes in minutes. So you can just make it into centimeter cube per minute or centimeter cube / minute. Alright, so part 2. So suggest the reason for the oil layer in the apparatus shown in figure 1.1. So why we want to add oil, right? Okay, you see on the apparatus, they have added an oil layer. So oil, right, they usually helps to create a barrier. It's very important that it creates a barrier to prevent oxygen from entering, because oxygen cannot penetrate the oil layer that easily. So what you have to do is to make sure that it is covered so that no oxygen can be going in, because you want to measure by the process of anaerobic respiration, so you cannot have oxygen involved in it. Alright, so that is the answer. So now you need to see, state one reason why no more carbon dioxide gas was produced after 35 minutes by the yeast in a sugar solution shown in figure 1.2. So why is this? It's because all glucose has been used up by the yeast cell. So meaning, you see, after 35 minutes roughly from here, you can see that it has become the same. So you need to know that the glucose has as a substrate, right, has already been used up. Once they've been used up, there will be no more anaerobic respiration occurring. No more carbon dioxide will be produced. So that is the result of this one. Part 4. The scientists repeated the investigation using yeast and the sugar solution at the temperature of 95 degrees. So explain why no carbon dioxide was produced at the temperature of 95 degrees. So it's very important to tell you that anaerobic respiration itself, it is involving a lot of enzymes. Okay. So this is a very big thing that you have to link. When you know that there is enzyme involved, right, you know that they cannot surpass at more than 70 degrees because it will denature. So as a result, you write your answer as enzyme will denature. Then what happened next? The active site changes shape because the glucose is unable to bind to it already. So it cannot really fit complementary. So then the changes shape will be not complementary for the substrate binding. So that is just it. It's just the effect of putting this enzyme at 95 degrees. What's going to happen? Alright. So part D, state one way in which humans use the carbon dioxide gas to produce by yeast cells. So this one, a very good example is bread making. So you know how bread can grow and expand is all because of the yeast and the anaerobic respiration that happen that releases the carbon dioxide out. And then part E, state the name of one gas other than carbon dioxide that contributes to enhanced greenhouse effect. So none other than methane. Okay. Alright, so let's move on to the next question. So question 2 is involving your coordination and nervous system. So coordination chapter. So I think it's chapter 14. So now they ask you to define the term sense organ. So it has been a frequent tested question. It's just basically testing you what is an organ basically. So you know an organ is comprising of a lot of tissues like that. So in that case for sense organ, right, I would say there's just a lot of receptor cells. Okay, receptor cells are very important because you need to have a lot of receptors, only that this can become a sense organ. So it can only detect stimuli. So you see, it helps detect stimuli from the surrounding environment because the stimuli will cause it to bind, release all of the the neurotransmitters and everything to bind on the vesicles that cause to produce a response in as result as you know that this stimuli has been provided. So that is the answer that you're supposed to give. So part B, figure 2.1 shows the structure of the eye. It also shows the pathway taken by nerve impulses which help bring about changes in the eye and the order of bring light to focus. So part 1, they ask you to draw a label line and a letter X on the figure 2.1 to identify a motor neuron. So motor neuron is always after it has been integrated into the brain and now the response is generated to the particular muscles. So you need to know that it's going to be after the brain, so that's why it's located here. And then one more thing that you need to know is that motor neuron always links to the effector later on, where it will produce what kind of response that it's supposed to produce, like it either contraction or it either becomes loosened, relaxes. So this is something like that, all right? And then part 2, describe how the events that occur at the synapse generate an impulse in the next neuron. So this is basically the synaptic transmission. So it's very important that you know this process because it holds a lot of marks. So you need to mention that neurotransmitters is released from the synaptic vesicles because there is impulse or stimulus has been detected. Therefore, the neurotransmitter is going to act like a messenger. All right, and then it will diffuse across the synaptic cleft. So remember that it shapes almost like this. Something like that, and then it has to go there. So this is the synaptic cleft. Alright, so then the neurotransmitter will bind to the receptors on the postsynaptic neuron. Then this will further continuously transmit the impulse that it has already made. Okay? Alright, let's look at the next one. So part 3, describe and explain the process of accommodation in the eye to view the near object. So when it places nearer, you can see that of course in a 4-mark response, the first thing very important is they want to test you whether you know what are the labelings are and what are involved. This can get you very important 1 mark here. So E is the ciliary muscles, K is the suspensory ligaments, and F is the lens. So what happens during accommodation of viewing a nearer object? First thing, the ciliary muscles will contract. Okay, and then the suspensory ligament will slack, or it can also be known as loosen. So it's always very important that you know that the ciliary muscles and the suspensory ligaments are antagonistic. It always has to be one of them contracting, another one relaxing. So it's the same that applies here. And the lens will become more convex because they are trying to pull the image closer to you and then causing more refraction. All right, more refraction, it means that it allows light to focus better onto the retina. Okay, all right, sorry. So that is the answer. So now you see, describe the distribution and function of rods and cones in the eye. So if you remember that there's one graph that shows the distribution, something like this, okay, and then it grows up. This is the distribution. So you need to identify that both rods and cones are found in the retina always, because that's where all the light will come to refract and then it goes back. So it has to be able to detect all of these colors that's been provided from what is the person seeing basically. And you need to know that no rods are found in the fovea, no rods and cones in the blind spot. So you can see that there's a gap between. This shows that this is the blind spot, where it has no cones, no rods, because that one is the location of the necessary nerve impulses to be transmitted to the brain. And light receptors and respond to light stimulus, that's definitely because you need to have these receptors that help to identify that, oh, this is a stimulus coming. And then of course rod will detect low intensity light, it's going to be a black and white image. And then of course the cones, they will detect high intensity light. And of course you remember cones as colors, so cones as colors. So very important, that's why you need the cones to be present there. Next one, red-green color blindness is a sex-linked characteristics. It is controlled by a gene on the X chromosome. There are 2 alternative versions of this gene, no color blindness XA and color blindness X smaller a. So when you realize that this is a sex-linked characteristic, it will involve either the male and the female. So you are also see that the color blindness is the recessive allele. So meaning if you have understand, you should have known that red-green color blindness is the X-linked sex-linked recessive trait. Alright, so you need to have both copies in the female to have color blindness for X smaller a, and only one copy of this XA for male is either XAXA or XAY. So state the term used to describe an alternative version of a gene. So alternative version is, you can see by the word A, you know this will be an allele, okay? And then state the genotype of a male with a color blindness. So it only needs one copy, and you know that male is X and Y chromosome, so you form an X smaller a Y. Alright, so that's very simple, 2 marks. Alright, now question 3a. Meiosis and mitosis are important processes in the life cycles of organisms. Figure 3.1 shows the life cycles of 2 different organisms. Organism 1 has a simple life cycle, and organism 2 have a complex life cycle. So it has stage A, which produces spores, and stage B that produces gametes, okay? And in the diagrams, the haploid number of chromosomes is represented by N. The diploid number of chromosomes is represented by 2N. Alright, so we have one is a simple life cycle. It could be the human. Let's take this as a human. And then after the organism 2 is by spores. So this is most likely a fungi. Alright, but now let's see the actual questions. So they say Table 3.1 shows the letter P or T in figure 3.1 and the type of nuclear division. So place a tick in each row to indicate the type of nuclear division that occurs at each letter from P to T. So let's go back and see from here. You can see that the adult one has become sperm and egg. So this is a diploid. And this has become a haploid, because gametes are always haploid number of chromosome, meaning this could be 46, this is 23, 23. So when they become a zygote is where they will add up together to become 46 again. So what's this process? This process is meiosis. Okay, then what is the next process at Q? Embryo is diploid. Okay, so after they form a zygote, they become a diploid. You know that this is, because it is 23 plus 23, you get 46. And then the zygote is just basically going to replicate all its cells to become embryo. So embryo will become the adult. So it is still the same thing and it's still multiplying. Therefore, it is mitosis. Alright, so because you are retaining the same 46 to 46. Okay, now you see for R. So for multicellular stage, it is 2N. Let's say it is 46 again, and you become spores is 23. So what does it undergo? Meiosis again. Okay, so because you are basically half the number to become a haploid is half of it. And then you have meeting S. Okay, so multicellular stage from N, they become female gamete N, and then male gamete N. Immediately the number of this is 23, this is also 23, this is also 23. So this is undergoing mitosis because it did not become 2N to N or other usually that one. So the only way is 2N to 2N or N to N. So this is mitosis. Then what happens at T? Zygote to embryo. Same thing for the previous one. If you have 46 here, embryo is also 46. So it is to diploid. So this undergoes mitosis. So that is the answer for this one. So state the name of process W shown in figure 3.1. So W, it is located at here. So sperm and egg, when they fuse together, it is usually called as fertilization. Alright, so fertilization is the answer. Now, an embryo contains stem cells. So you need to always know that stem cells is always unspecialized cells, and they can eventually differentiate to become specialized cells. So stem cells are unspecialized cells that divide by mitosis because the number of 2N to 2N is still the same. That can become specialized for specific function. It's always stem cells acting as like, I can become anything. And when I become anything, I become more specifically the specialized type. And then most body cells in organism contain the same gene, but many genes in the particular cells are not expressed. So it's not showing out. So this is usually the recessive. So, or you are a carrier, where you don't explicitly explicitly show that you have this, so it's expressed. Because the cell only makes a specific protein it only needs. When it has produced a specific protein, it doesn't need the other one already. Alright? So part B, state the events of the life cycle diagram of organism 1 that will not be present in a life cycle diagram for asexual reproduction. So first thing, it definitely undergoes meiosis. Then it forms the gametes. Then the male and female gametes will fuse to form a nuclei, and then a zygote is formed. So this is basically how fertilization has occurred. So now, we come to question 4, which is plants. And some algae are single-cell organisms that can photosynthesize. Their cells contain the pigment chlorophyll. Alright. So state the name of one mineral ion that is needed to make chlorophyll. So the one that makes chlorophyll is always the magnesium ion. So this is need to be you need to know this, because these are very important nutrients, that the micronutrients that the plant needs in order to produce a certain part of pigments for its growth. So part B, a student investigated the effect of light on photosynthesis and using hydrogen carbonate indicator solution. So remember, a hydrogen carbonate indicator is PM ROI, from lower CO2 to higher CO2. So carbon dioxide is an acidic gas. Okay, so the two test tubes were set up at the same time. Both contain algae and hydrogen carbonate. So at the start of the investigation, the hydrogen carbonate indicator was red in both test tubes. So initially it was this. And then one test tube is placed in the dark and one test tube is placed in the light. So after 20 minutes, the contents of the test tube in the dark were yellow and the contents of the test tube in the light were purple. So explain these results. So of course, first thing we mentioned that there's no photosynthesis happen in the dark, because they already mentioned that you need light in order to photosynthesize. And then CO2 is needed for photosynthesis to occur. So it turns purple. Okay, first thing we see, it turns purple because the pH increases as the CO2 concentration drops. So you know that CO2 concentration is acidic. So when it becomes lower, it means that the pH will go up or become more basic. Okay, and then with the yellow result, it indicates that algae will respire both the light and dark. So it's, remember that photosynthesis only happens at daytime, and then for respiration, it occurs both day and night. And CO2 is given out as a result because it is a product of the respiration. Therefore, it causes a decrease in pH. It becomes more acidic. So that's why it turns yellow, because it has high carbon dioxide concentration. Alright, so if you have any question with this, again, feel free to ask in the comment section again. Okay, now, so part 3 is figure 4.1, is a graph showing the effect of light intensity on the rate of photosynthesis. So they ask you to label what is X and what is Y. So it's very important that for you just separate this. For X, right, it's always responding to the X-axis. So light intensity will be your answer. But for Y, right, it can be different answer, but it cannot be light intensity again. So it can be either temperature, because temperature can differentiate. Either it's going to be this kind of graph that I show you, or it's going to be this kind, or it's going to be like this. So it can be a difference in temperature. And also it could be also CO2 concentration, because when you have lower CO2 concentration, it means your rate of photosynthesis is lesser, because less carbon dioxide is needed, is consumed to undergo photosynthesis. So you need to have high amount so that it can maximize its rate. And then another one, you can say the number of chloroplasts. The more chloroplasts it has, then of course the more photosynthesis it can take place. Something like this. Alright. Next one, so starch and sucrose are made by plants after photosynthesis. So these are the products. So state the name of the tissue that transports. So tissue could be either xylem or it could be the phloem. And then they transport sucrose around the plant, and name one other biological molecule that is transported in this structure. So first of all, it will be the phloem, and the biological molecule is amino acid. If I ask for xylem, so xylem will be the water and the mineral ion. So now, describe how starch is broken down in the human digestive system so that it can be absorbed into the blood. So firstly, you must know that whatever we have eaten, right, is always in the form of food. Let's say it could be rice. So rice is a starchy food. So when you when rice is entered into your mouth, right, first thing, it won't be straight away swallowed in, right? So first thing, it will try to break down by using mechanical or physical digestion. So mainly what causes it to become more, I would say the surface area has become larger, where it's because of your teeth biting, grinding, and crushing it. So it kind of just increases the surface area, so the enzymes can act a bit more efficiently. So the next, you know that is chemical digestion, where it breaks insoluble to soluble molecules. So you know that you give this as a form of a definition. And then so your salivary glands will secrete your amylase, and then amylase is an enzyme. So they will break down from starch to maltose, and then once it travels into the small intestine, you meet the small intestine enzymes. So one of them will be maltase. Maltase is secreted to break down glucose in a basic pH, meaning like about more than 7. So that's considered as basic. So question 5, figure 5.1 is a photograph of a koala. Koalas are marsupial mammals that give birth to offspring that are incompletely developed. The offspring develops further for several months inside the pouch of the mother. So state one feature that identifies koala as a mammal. So mammals typically is a very easy way to find, is that first thing, they have fur, or you can say they have external ears or pinna, alright? And then for B, mammals use sexual reproduction to produce offspring, and now they ask you for the advantages of sexual reproduction. I always, if you can't really remember this, it's best that you compare these advantages to us as human. You can see that everywhere you go, humans look different. So this, what does everybody look different mean? It creates an increase in the genetic variation, because there's more people who look more different from each other. So what happens if they look more different from each other? It means that they have a different set of a DNA sequence. Therefore, when this happens, they can express different characteristics from the alleles that it provides. And then one more thing, since you know that it's a different sequence, everybody has a different sequence of the DNA, therefore they can adapt and they won't die easily by a disease. Because if you have the same genetic sequence as the other person, right, you get the same disease, the other person also gets the same disease, and then if they die, you also die, because you got infected by the same way, you have the same genetic coding, alright? So there's nothing unique there, alright? So that's how you link it. So next one, part C. Figure 5.2 is a diagram part of the human female reproductive system viewed from the side view of the body. So you must be able to know how to identify this from the side view, of course. So complete table 5.1 by writing the letters that identify the structures in figure 5.2 and the names of the structures and the functions. So first thing is cervix. The cervix is located, it's like a C shape, you can see. So this is M. So what happens there is that it dilates during the birth process to allow the fetus to come out from the mother's reproductive system. And then the next one is the ovary. The ovary is very indicated, it's like an O shape. So this is an L. So it releases mainly estrogen hormones. And then the site of fertilization is always at J. So it could be the oviduct or it could be the fallopian tubes. These are also accepted. And then the site of implantation is at P. So P is here. So this indicates that this is the uterus wall or the uterus lining. Alright, next one. So pregnant human females have a placenta. So placenta is the one that links from the mother to the fetus. Now they are asking you, what is the function of the placenta in the humans? So since you know that it's between mother to fetus, so you know that gas exchange can occur because the carbon dioxide has to go back to the mother, and then the oxygen is transferred to the fetus. And then what else? You need to know that it can transport dissolved nutrients, important ones, and transfer of excretory products, and also transfer antibody for passive immunity. Alright, so all of these are important for allowing for the baby to not have any problems the moment that it comes out from the reproductive system. And then it's all mainly to gain passive immunity. Okay, So part 2, the placenta is connected to an amniotic sac. So state 2 function of the amniotic fluid, okay, that is found in the amniotic sac. So amniotic fluid is just like it's a fluid that, you know, it has a balanced osmolarity gradient, that everything is proper, it is sufficient, it's sterile, it supports the fetus, and then maintain the temperature of the fetus, protect the fetus from mechanical shock, and some more as allow movement of the fetus. So it creates that sort because it's almost like a lubricant. It allows it to move easily. Alright. Then now you see question 6. Complete the sentence about human teeth. So the process of taking food into the mouth is known as ingestion. So the flat, sharp-edged teeth in the front of mouth is called as incisors, are used for biting off pieces of food. The molars and premolars are the large teeth towards the back of the mouth that are used for chewing the food. So figure 6.1 is a diagram of villi. So now they want you to label, okay? I think if I'm not mistaken, is to draw labels and label line on figure 6.1 to identify a capillary and a lacteal. So they ask you for 2 things for 1 mark. So you need to always use a straight line and not an arrowhead. This is not acceptable, and you label. So capillary can be here. It's really surrounding the epithelium, and then the lacteal is found in the very inside of the villus. So now, state the part of the digestive system where the villi is located. This is very simple. It will be at the small intestine. And state the function of the lacteal. The lacteal is to absorb and transport fatty acids, so to other parts of the body, and describe the pathway taken by the products of protein digestion from the villi to the liver. So it's basically from the small intestine after it has broken down into smaller pieces by the enzymes, the trypsin, and many more, pepsin. So how is it going to go? Amino acids will move to the capillaries first. So capillaries will be sent into the bloodstream already. Okay? And then most importantly, to the liver, it has to meet with the hepatic portal vein. So this is the last site where it has this, okay? So this is the end of the paper. So I hope everything's okay. This is a rather easier paper than May-June 2023 paper 4 variant 2, because it's a bit more vast. It tested really like what you're supposed to expect like that. So overall, if I check with the threshold, this is actually quite high. It's about 172 total out of 200. So again, never mind, but in case you have any questions, again, feel free to comment in the section below. Thank you so much for watching again. Good luck and all the best. Bye bye.